All right. Um, so we very quickly, but we went through the different types of mutations. So there are several broad categories of phenotypic effects of mutations. So we've got sort of the immediate effect on the protein. So we haven't really talked about that yet, although that's what you all were doing in your packet when you were changing your amino acids, putting in stop codons and whatnot. You were affecting the actual protein. Once you get the protein, then this altered protein, there's a variety of ways that the phenotypic effects of that mutation can show up. So if you recall, at some point, we define wild type versus mutant. Wild type is basically the normal or the most common in a population. So usually we talk about this in genetics class. It's related to usually to fruit flies, and we talk about the wild type fruit flies, but really you could use it to refer to anything. So we've got the wild type mutation, and then we've got, or I'm sorry, wild type phenotype, and then we've got the mutant type. So a forward mutation is where your wild type is converted into the mutant. And the mutant may or may not have a negative effect on the individual. It just depends on the actual gene itself. Reverse is where we go from mutant to wild type. So most of these are really just sort of vocabulary words. There's not a lot going on here. A loss of function mutation is where essentially the protein, usually the protein is not produced, or a functional version is not produced. So you basically have a dysfunctional protein here. Gain of function mutation is basically going to restore the normal protein function. A conditional mutation is just like it sounds, it's a mutation that only shows its effects under certain conditions. So this is usually going to be some sort of environmental effect. So for example, we were just talking about the lac operon. If your cell is normally in presence of glucose, using glucose for respiration, you won't know that there is a mutation in that lac operon until it attempts to use lactose. So we're only seeing that mutation under the right set of conditions for whatever that gene is doing. And then lethal mutation means that it's lethal, makes the organism die. Not rocket science there. So these are the effects on the actual phenotype of the individual. But these are going to be enacted by the actual changes to the protein itself. So we've got a couple of different ways that we can actually affect the protein structure. So the first one is a missense. This is where you substitute one amino acid for a different one. Now this one is not always detrimental. You can have something called a neutral mutation. This is where you substitute a different amino acid, but you cause no change in the function of your protein. In other words, that amino acid substitution doesn't cause any shape change or functionality difference in your protein. So if you remember from our discussion on proteins, we talked about how our amino acids have different chemical properties based on their R group. So they might be acidic, they might be basic, they might be polar, non-polar. So if you substitute, if you have a missense mutation that, let's say, substitutes a polar amino acid for another polar amino acid, you may not actually change the functionality of that protein. You may not actually change the overall structure. If this new polar amino acid is behaving in the same way as the previous one, then you can get a mutation, a neutral mutation. So meaning that you have made a substitution, you have changed the amino acid, but it doesn't affect that protein's ability to function. You can also have a nonsense mutation. This is where we change from an actual amino acid coded by our codon into a nonsense or a stop codon. So we have 3 different stop codons. If you introduce a stop codon, then we are going to truncate our protein. So our protein is going to be shorter than expected. And this one is always going to cause a dysfunctional protein. So this one's always going to affect your phenotype. So you are going to get no functional protein out of this one. Because once that stop codon is hit, remember that causes the ribosome to completely fall apart and nothing else gets added after it. I don't remember if it was the 2 of you, but a lot of times on those packets, when I get them back, we've got stop codon and then more amino acids and then stop codon. That stop codon is like the end of the sentence at the end of a paragraph. You get nothing else added. So you can't get more amino acids added after that stop codon. So it's just going to be a little bit shorter. And then we can have a silent mutation. So this happens when you have a mutation, but it doesn't actually change. This should actually say no amino acid change, not codon. It doesn't change the amino acid. So in chapter 11, when we were talking about the genetic code, we were talking about redundancy. And this was related to wobble, which is where your third base in your codon can change without causing a change in the amino acid. So generally, not all of them, but most of them have multiple codons that code for the same amino acid. For many of them, they have 4 codons. And so you can change that last the last base pair, the last nucleotide, without actually changing the protein sequence. So down on the bottom, this is just a diagram to show each of these. So if our original one codes for a serine here, our missense is switching out that amino acid, so changing the amino acid. Nonsense, we get a stop, so we don't get anything else added afterwards. And then the last one is showing that silent mutation, where we get a substitution, A for G, and then that still codes for serine. So we don't get a new amino acid there. A to G, is that a transition or a transversion?
Transition.
Purine to purine? That should be a transversion. Nope, you're right. Nope, 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 hold on. Transition, you are correct. Very good. I have to always like go through all of them. I can't remember at the moment how I remember those. but because they sound like the name sounds the same. Transition, transversion, you're still switching something. Okay. All right, so causes of mutation. So there's two big causes. One is replication errors. So this is where DNA polymerase puts in the wrong base. It could be this one is going to be the transition or transversion. That results from incorrect replication. So we talked about how DNA polymerase has proofreading abilities. So our main DNA polymerase 3, as it's going along, it can actually register if it is put in a mismatch base. And if it has, it can pause, back up, swap it out. We also talked about how DNA polymerase 1 can go behind DNA polymerase 3, recognize when bases are incorrectly paired, excise the particular nucleotide, and put the correct one in. We can also get spontaneous changes, so due to environmental factors. One of the things that it can do is affect the functional groups that are attached to the DNA itself. So in chapter 12, the part that I skipped out, you can actually control gene regulation by affecting the overall DNA structure. You can also cause mutations by affecting the overall structure. So we can lose a purine. So if you basically delete a purine out of a sequence, we can also cause changes to the amino groups that are on our amino acids. So we can also affect the physical structure of the protein by inducing chemical changes. If we're looking at DNA, those chemical changes are going to be changes to the DNA sequence itself. All right, so you all probably know this already, but there are different chemicals that can cause damage to the DNA. So they can cause structural damage, so interrupting covalent bonds. So some of the big ones are exposure to mutagens and carcinogens. These can be chemical carcinogens. They can be UV light, smoke inhalation, asbestos, things like that. Radiation can also cause mutations to occur. So specifically, radiation causes your something called thymine dimers. So there's a I don't know if I have a picture. There's a picture later, I think, of a thymine dimer. Basically, what happens is instead of being paired across with the adenine, we get an extra covalent bond sideways, so binding T to T. And then this is going to disrupt the ability to replicate or transcribe that particular sequence. All right, so we talked about, when we were talking about DNA structure, we talked about how our purines always bind with a pyrimidine. And for our base pairs, we get adenine bound to thymine and guanine bound to cytosine. Across the double-stranded helix, those sequences are paired and held together by hydrogen bonding. So in a correctly paired situation, adenine binds with thymine by 2 hydrogen bonds. Guanine binds to cytosine by 3 hydrogen bonds. If you have non-standard base pairing, what you get is basically, obviously you get incorrectly paired bases, but you can still get that double strand held together because we have the right components for hydrogen bonding. You can still just get abnormal hydrogen bonding. So even though we are inserting an incorrect base pair, you can still get relatively correct base pairing across that double strand. So as far as the actual overall structure of DNA, those mutations are not necessarily a big deal. But if you are expressing genes from those sequences, then you can get those nonsense, missense, silent, and neutral mutations. All right, so 2 more ways that we can introduce mutations. One is through something called strand slippage. This happens usually when you have a run of the same nucleotide. Basically, because in this example, you've got A, A. What can happen is a piece of the DNA strand can actually bulge out. And then hydrogen bonding occur through the next nucleotide in the sequence. So we've got examples here. The example 1 is the top strand. So as synthesis is occurring, you get the top strand slipping out and producing a little bump. And then the result of that is that now when we replicate it, we insert an extra A. So the next time those strands pull apart and replicate, then we have inserted that extra A. So that is going to be a result of an insertion. So slippage on the new strand, that's going to lead to inserting a base pair. If you get slippage on the template strand, so basically the same thing happens except that the template strand moves, what you're going to have is the template strand bulging out. So then on the new synthesized strand, you're actually going to lose a base pair. So for this one, this is where we're going to get the deletion. So instead of having 5 on the original strand, we're only going to have 4 on that daughter strand. Are there any questions? Usually this one's pretty straightforward, so kind of going fast, but do you all have any questions? All 2 of you? No? Okay. It's okay if you don't. All right, so we can also have insertions and deletions from unequal crossing over. We talked about this when we were talking about chromosome insertions and deletions. Remember we said that this was a while ago, but if you delete something from one strand, you're probably going to duplicate it on another because crossing over is not going to remove that sequence, it's just moving the sequence. So the same thing can happen for short segments of sequence. So we talked about it already for whole chunks of chromosomes, but same thing can happen at more of a localized level, and you get just a small number repeated or deleted instead of a whole big chunk of the chromosome. Sorry, I've got my kids blowing up my text message thread. They like seem to forget that I work sometimes. Like, go away. If I ignore my youngest, though, he'll call me. So I can't ignore him forever. My oldest will give up, but my younger one will just keep bothering me. Okay. Which is good because I need to keep better eye on the younger one. Okay. So one of the handouts I think that I gave you, I hope, I think I gave you a summary of the different types of mutations. Maybe I didn't give you the DNA repair. It would be on that stack that you have right there, DJ. On the back of one of those. Did I give you? No, that's control. Oh, maybe I didn't. Okay. There's not too many. I don't think maybe I didn't give you a summary of the mutations. There are a couple of different ways that you can repair DNA within the cell. So if the cell recognizes that there's a problem, there's a couple of different things that the cell can do. So we talked about that proofreading activity that DNA polymerase has. And then the cell can also identify if we have a sort of an environmentally induced problem. So in general, DNA replication is pretty accurate. The proofreading mechanisms are pretty accurate, but sometimes things kind of get through anyway. And then of course we've got external mutations the cell really cannot control. So all of these kind of use the same type of idea. So mismatch repair, base excision, and nucleotide excision repair, these kind of all work roughly the same way. So if the cell recognizes, like if DNA polymerase 1 is going through and recognizes that there's a mismatched base, basically it brings in a set of enzymes that are going to pull out the mismatch and replace it. So up here we've got guanine and thymine paired incorrectly. So the DNA polymerase can go through, recognize, because the structure, there's a little bit of a structure issue there because we've got the incorrect pair, recognize that there's an issue. So when that happens, we get enzymes that come in that basically make a little cut in the sequence, pull out that base pair and replace it with the correct one. Excuse me. So we basically do like just a little tiny bit of DNA replication there. Just pull out the base pair that is incorrectly matched, place in the correct one. It is possible here to replace the wrong side. So for example, we've got the new strand here. The new strand is the problem. If the new strand doesn't get replaced and the old strand gets replaced instead, then we've just sort of proliferated that repair mechanism. So up at the top, this one is going to be our mismatch. This one, the next one, the next two actually are going to be our base excision and nucleotide excision. So we can also have direct repair. So direct repair is going to happen when your nucleotide structure or your functional groups are changed in your on your DNA nucleotide. So this can happen in one of two ways. You can do a base excision, so pull it out, kind of like mismatch repair, pull out the actual base, or the cell can also alter the base itself. So I mentioned a minute ago in chapter 12, we skipped over the eukaryotic controls. Basically, one of the ways that transcription can be regulated is by methylating guanines. So adding a functional group to a guanine, which essentially changes the function of that guanine molecule. So the cell can actually remove that methyl, or if we've got a chemical change to one of these, the cell can repair a chemical change as well, by either removing the nucleotide, replacing it with an unaltered one, or actually physically changing that nucleotide. So we just talked about mismatch repair on the previous. This is where you're specifically where your sequence is improperly matched during DNA replication, so you don't insert the complement. Then there's also just sort of general nucleotide excision repair. This one is a little bit more general. So if something happens besides just a single nucleotide mismatch, say you have a couple of nucleotides or a region of DNA that's actually damaged, the nucleotide excision basically acts as scissors to remove a short section of DNA. So depending on how big this is, if it's just a single nucleotide, we just have mismatch repair. DNA polymerase 1 can do that. If it's larger, if you have a big problem or a multiple nucleotide problem, we talked about those thymine dimers a minute ago where thymines bind across. You can remove those, and then DNA polymerase comes back in, fills in the gaps, and then DNA ligase has to come behind and connect the nucleotides into the rest of the strand. All right, so just a quick example. Sometimes the inability to repair DNA causes genetic diseases. So the example here is called xeroderma pigmentosa. This is basically where normally if you have UV damage to any of your skin cells, the cell will recognize it and can repair that damage. But xeroderma pigmentosa is essentially regions of the skin where there's a faulty repair mechanism. So the cause of cancer is actually not necessarily directly the mutation itself, but it is the inability to repair the mutation. So I would not suggest Googling this. This is the textbook image. It's a very nice image. I would not suggest Googling this and looking at some of the other images. I was looking for, I couldn't find this image when I was editing this PowerPoint, and I couldn't find the original one. And so I was like, oh, I'll just Google it and put in another one. Changed my mind really quickly, worked harder to find this one. So basically, when you don't have when your cells don't have the ability to repair DNA, you have a greater predisposition for skin cancer. So characteristics of this disease are freckle-like spots on the skin. So these are basically more intense than freckles. Extra sensitivity to light, and then predisposition to skin cancer, because you don't have that repair mechanism that is constantly going behind the UV damage and repairing it.
And then in your body, like if your DNA doesn't do its job, like it stays that way permanently, there's no way to fix that.
No, so if it is, so if you have let's say you have a mutation for one of the enzymes that is supposed to do the repair, then it's like any other genetic disease or disorder. You're just kind of stuck with it. So once the mutation is introduced, then it's just going to be proliferated. So then it kind of goes back to the gamete versus somatic mutation. So if this is a mutation that has happened in your gametes, you pass it on to the next generation, then all of the cells might have the faulty repair mechanism. If it is something where, like, let's say that maybe UV radiation affected that gene, so the DNA repair gene, then it may not get passed on to your kids. But wherever that faulty gene is, any cells that come from that cell line are going to have that mutation. So in that sense, it's kind of like any other genetic disorder where you have a mutation causing that disorder. You're basically stuck with it. So I mentioned, I actually can't remember. I know I talked about it with Jacob when Jacob came in, about the CRISPR lab. There are 2 approved CRISPR editing mechanisms, like FDA approved treatments for sickle cell anemia. So normally with a genetic disorder like sickle cell, you just have it. And the only thing you can do is manage the symptoms, manage the phenotype for it. But with CRISPR, because you can edit in live cells, they've actually developed a gene editing sort of fix for sickle cell. So if you can get the CRISPR edits into your bone marrow cells that are producing your red blood cells, then you can potentially fix the disease. But right now, it's very, very expensive, but you could potentially edit sickle cell. At the moment, all the other diseases, there's no fix for it. So all you can do is manage the symptoms. Does that answer your question?
Yeah.
If you're looking at the online slides, there's just a list of some other faulty repair disorders. This is the end of 13. So 13 is quick, 12 is quick. I decided I didn't want to do all the 12. I just wasn't feeling it. I do want to move on to 18. I know we're kind of jumping around doing a lot of stuff today, but I do want to move on to 18 and at least get started with chapter 18.
And that's the last chapter?
That's the last chapter. The only thing we might do, if we have time, is go back to 14 and do some DNA sequencing. And there was one other topic in chapter 14 that we didn't get to as far as technology goes. So we might go back and do that. Probably not, but we might. We'll just see.